once again on the 5th week of this course. So, today we are into lecture 24th, and this lecture will be about the recognition elements, specifically scFvs. Okay. What are scFvs? So, in terms of definition, if you look at it, scFvs, what it is. Schematic representation of a full-length monoclonal antibody and a single chain variable domain FV fragment. This is called scFv. Okay. So, I told you that most of these antibodies have variable, heavy chain and light chain. So, this is the variable region. You look at it in this picture, this is the variable region, and heavy chain which is shown in blue, light chain which are shown in red, and this particular part is called scFv. So, if you remember in the last class I was mentioning that each one of these parts are coded by specific genes or DNA sequences. Okay. Now, if you could isolate that particular sequence of DNA which codes for this particular part and you express it in some, say, bacteriophage protein, surface protein, then on the top of that particular animal. So, it's just visualize the situation. So, you have a DNA for this particular part and you couple this DNA with something which will be expressed on a surface protein of a specific cell. Now, what will happen on the surface of that particular cell? You will see the expression of these kind of molecules. So, this is what is scFvs, which is single chain variable domain FV fragments. Okay, single chain variable domain FV fragment, and can be engineered into a multivalent species, diabody, tribody or tetrabody. So, you have these are the dibodies, 2 of them, tribodies and tetrabodies were 4 of them. Tetra, di, tri, tetra. You could have even have more of them configuration, but these are the common configurations. So, do not think that these are the only configuration. Now, scFvs has a whole range of usage if you look at it. So, single chain variable domain, which is represented by FV variable domain fragments, okay, are powerful tool in research and clinical setting owing to the better pharmacokinetic properties compared to the parent monoclonal antibodies and the relative ease of producing them in large quantities at low cost. Unlike monoclonal antibody production, where we talk about the hybridoma technique, phage display technique, the production of this single chain variable domain fragment is far more economical because you are just taking a small part of the DNA, you are coupling it with a surface protein of the bacteriophage, and you are producing them in a large number. Okay. So, though they offer several advantages, they suffer from lower binding affinity. Why there is a lower binding affinity? Because think of it, when we look at the structure, in the normal case of a monoclonal antibody, this whole thing is going and kind of there are multiple binding site where it is binding to the host. Contrary to that, when we are only expressing this much part, so automatically your binding area is reduced. So, binding may happen at a higher affinity, but then you need a lot of this. So, it is kind of a trade off. If you have a lot of these, then of course, the binding will happen much better. But if you have a lesser amount, the binding will be lesser because your site of contact, site of bindings are less, right? Because these are very specific site where it binds. It is not that everywhere they will be binding. So, if this whole antibody as a monoclonal antibody has this many binding site, then this will have only this much binding site. So, try to realize in a physical frame of your mind why scFvs have a lesser binding. Though they offer several advantages, they suffer from lower binding affinity and rapid clearance from circulation, which limits their therapeutic potential. However, these fragments can be genetically modified to enhance desirable properties such as multivalency, high target retention, and slower blood clearance. And as such, a variety of scFv format have been generated. scFv can be administered by systemic injection for diagnostic and therapeutic purpose. They can be expressed in vivo through viral vectors in instances where large infection rates and sustenance of high level of antibody is required. scFvs have found application as tools for in vivo loss of function studies and inactivation of specific protein domains, diagnostic imaging, tumor therapy, and treatment of neurodegenerative and infectious diseases. So, now having said this, it must be clear why there are more preference of these dibodies, tribodies, tetrabodies, likewise. And I will take you back to this picture, what I was telling you in the last class. Look, these are the ones. All these different kind of configurations which are present there, these are all getting generated because of, so look at it, where they are binding. So, their binding sites are out here with the cytokines where they are binding. Again, here the cytokines binding, here again the TNF alpha or tumor necrosis factor binding. So, you see the number of binding sites become much lesser, but they could be genetically modified, as it's being mentioned, that they could be genetically altered further in order to slower their blood clearance, enhance the desirable properties like multivalency, high target retention, and these ease of genetic modification of these fragments makes them a popular choice for futuristic therapeutic potential, and they are far more specific as compared to monoclonal antibodies. So, if we remember back in 80s, when you used to get the polyclonal antibodies, you are very happy. Then came the whole era of mass production of monoclonal antibodies. So, things started to change and everybody was asking, okay, let's get a monoclonal antibodies for immunocytochemistry, immunohistochemistry. Followed by that, there is a next era which came, scFvs. You get even much more. And you started working on the nanobodies. So, nowadays I see people who work on GPCR and all other, they work on these nanobodies, even much more smaller fragment. They're very specific. But whenever these kind of things happen, always remember, in order to recognize them, the kind of fluorescence signature you need, the clear imaging you need, those are also equally challenging, because when you are picking up such low signals, you are also picking up a lot of noise. In practical life, while these are extremely potent and potentially path-changing discoveries, yet they come with their own set of challenges in terms of signal to noise ratio, the binding, and further detection using other technologies. Okay, so this is overall how the scFvs looks like. From here we move to the next, called aptamers. So, before I move on to aptamer, if you look at the phage display based on bifunctional sensors, so these are those sensors when we talk about, these are the kind of detection level what is happening. So, you have the gold binding peptides, on the gold binding you have the gold which is getting bound on it, then you have the antibodies which are binding on top of it, and then you do a display, and this is used for Bacillus anthracis, phage display of scFvs and many copies of the gold binding peptide on its surface by fusion expression with P3 and P8, allowing B anthracis recognition and signal amplification. Detection sensitivity of bifunctional phage immunoassay with silver enhancement and phage ELISA of Bacillus anthracis spores. Specificity of bifunctional phage display immunoassay. So, these are the practical applications where Bacillus anthracis is being detected using these kind of technologies what we have just covered. So, now from here I move to aptamers. So, this class is further from scFvs, now move to the aptamers part. Aptamers are single-stranded oligonucleotide. So, now we are talking about nucleic acids that fold into a defined architecture and binds to targets such as proteins. So, aptamers are sequence of oligonucleotides which binds to proteins. Okay. So, now we are venturing into nucleic acid protein binding. So, here nucleic acid are recognizing specific proteins. In binding proteins, they often inhibit protein-protein interaction and thereby elicit therapeutic effects such as antagonism. Aptamer therapy prevents the protein-to-protein interaction, protein-to-protein signaling, thereby it elicits therapeutic effect such as antagonism. Aptamers are discovered using SELECT's systemic evolution of ligands by exponential enrichment. Systemic evolution of ligands by exponential enrichment, a directed in vitro evolution technique in which large libraries of degenerate oligonucleotides are iteratively and alternatively partitioned for target binding. They are then amplified enzymatically until functional sequences are identified by the sequencing of the cloned individual. So, this is what is aptamer technology is about. For most therapeutic purpose, aptamers are truncated to reduce synthesis cost, modified at the sugars and capped at their termini to increase nuclease resistance, and conjugated to polyethylene glycol or another entity to reduce renal filtration rate. So, there are nucleus all over the body which can chop off the nucleic acid because it breaks the nucleic acid. So, there are techniques by which you prevent the nuclease activity and you couple it with polyethylene glycol or other entity to reduce the renal filtration. So, you are increasing the retaining time within the body. The first aptamer approved for therapeutic application was pegaptinab sodium, which was approved in the year 2004 by US Food and Drug Administration for macular degeneration. This is the first aptamer which has been approved. 8 other aptamers are currently undergoing clinical evaluation for various hematology, oncology, ocular and inflammatory indications. Aptamers are ultimately chemically synthesized in a readily scalable process, which specific conjugation points are introduced into the defined stereochemistry. Unlike some protein therapeutics, aptamers do not elicit antibodies. Remember this. And because aptamer generally contain sugar modified at their 2' position, toll-like receptor mediated innate immune response are also abrogated. So, when we talk about TLR or toll-like receptors, in short these toll-like receptors are innate immune system. That means TLR receptors are present across the surface of your body, across the orifices of your body, across every entry port. It's like a toll. You all have heard about the word called toll. You have to pay toll while you are traveling on the road. Okay, toll tax. It is exactly the same word which has been used in immunology. In the innate immunity, your surface of body has lot of toll-like receptors. So, whenever you are introducing anything into your body, the first line of defense is the toll-like receptor. They prevent the stuff to get into your body. And there are a lot of efforts which have been made, which are still underway to prevent the entry of AIDS virus by activating the toll-like receptors. Okay. And toll-like receptors are the ones which elicit a huge amount of innate immune response, but it has been seen by modifying the sugar at the 2' position, toll-like receptor mediated innate immune response are also abrogated. So it means you are giving the aptamers a free hand to act onto the specific protein and abrogate their interaction with another protein. So in a way, you are striking at their signaling platform, right? As aptamers are oligonucleotides, they can be readily assembled into a supramolecular multi-component structure using hybridization. These are pretty small structure. They are just some small thread-like nucleic threads. Okay, so they could be made into a complex supramolecular assemblies as a therapeutic agent. Owing to the fact that binding to appropriate cell surface target can lead to internalization, aptamer can also be used to deliver therapeutic cargoes such as a small interfering RNA. Further, supramolecular assembly, as I mentioned, of aptamer are delivery agent have already been demonstrated in vivo and may prove the way for further therapeutic strategies with the modality in the future. So, if we look at it in terms of aptamers, so the aptamers are basically single-stranded oligonucleotide and they fold into a defined architecture and bind to targets such as proteins. So, they can fold something like structures like this, they could have structure like this, and they have a very unique geometry in 3 dimension and they form supramolecular assemblies. They can bind to any of these proteins, and molecular manipulation of aptamers is far more easier versus molecular manipulation of bigger protein molecules like antibodies and all. So, if we look at it in terms of the recognition elements, we started with toxins. So, if I go back into the recognition elements where we started our journey, as we are almost about to complete all the recognition elements, what we talked about, it is worth visiting all the recognition elements here. So, these are our vegetative cells, domain, spores, and so on and so forth. And these are those elements: toxins, DNA, surface proteins, DPA, others. And now, if you look at all the recognition elements out here, this is what we covered: monoclonal antibodies, scFvs. I did not talk much about the peptides because we'll talk about it a little bit. These are simple, you can say 2 to 20 amino acid chains, small peptides are there. Then aptamer, which is a nucleic acid or nucleotide, and then the fast display using scFvs. So, the reason I devoted these 4 classes for these recognition element is, first of all, you have to understand the basic molecular architecture of recognition elements. I just put RE. Second, their interaction mechanism. So, for example, in case of protein, they interact differently, like MABs, scFvs. These are protein fragment versus aptamer. Second, how these could be produced in large scale, and further how these could be quantified, the interaction. That's why understanding of these are important. While we are making a biosensor, these are the key element which will recognize these targets. And when you talk about biosensors, once they recognize this target, you need a way to measure these interactions using either QCM, SPR, LFIA, PCR, nanomaterials, and so on and so forth. And the signal output of all these different interactions could be either electrochemical, SPR angle, piezoelectric, Raman, EI, MS, DNA amplification, color, fluorescence. So that's why you have to understand these recognition elements as efficiently as the target elements. If you understand both of them, you can design a better biosensors at your disposal. Because with this, I will move on to in the next class with the SPR, which is the signal generation device. Okay. So far, what we try to do, we talk about monoclonal antibodies, where we talk about the fundamental of monoclonal antibody. Then we talked about the difference between monoclonal antibody and polyclonal antibody. Then we talked about the two key techniques of production of monoclonal antibody, which includes hybridoma technology, phage display technology, and then we talk about B cells, using B cells and protein sequencing technology. Then we talked about scFvs and how there's a smaller units of variable heavy chain and all could be utilized, and how they could be made more potent so that they could be retained in the body and they are not neutralized by the nucleases, all the molecular manipulations what could be done on them. Thereafter, we talked about the aptamers and how they are being used, and in between we talked about the phage display of scFvs and how they are being isolated, they are being screened, and finally we talked about different techniques of quantification of these kind of interactions. So with this, I will close the 24th class, and the next class, in the 25th class, we will talk about the SPR. Thank you.